Hey guys, so this video is really to talk about mitosis and show you how you should learn it. I've noticed that students and myself, we only can learn mitosis if we go through a painful process of drawing it over and over again until it's embedded in your skull. And so there is no other way around it, so we're gonna do it. I use a whiteboard, you can use a piece of paper. So you will start by just realizing that you need to start in G2, the phase being G2, that's the late interphase, right? So when you start in G2, your cell has the nucleus, and the nucleus is always with a nuclear pore, right? So you can represent it more or less open, but the nuclear pore usually allow DNA to reach out. And during that phase, the DNA is replicated. So in that case, I'm only going to be making one chromosome here. You can notice it has two sister chromatids because we passed the replication, right? So now the molecular mechanisms are already in place. So you have the centrosomes that are those giant structure that I make two rectangles for those. And those structures here are what will direct mitosis. So I usually draw them this way, the centrosome, with a centriole and some of the proteins. So importantly, when you go into mitosis, because this is still interphase, when you go into mitosis, three things happens during prophase. And prophase, prometaphase, I'm going to lump those two together, right? It's more prometaphase than metaphase. Prometaphase is kind of an in-betweener. So let's just focus one at a time. We're just going to do prophase for now. Prophase, there are three different steps that are extremely important in prophase, and you need to remember those. So if we make our cells for prophase, the first thing that happens in prophase is the nuclear envelope disappears. So the way I like to represent that is to make a dotted nucleus, right? And the nuclear envelope disappearing is going to allow the centrosomes to reach the chromosomes. Not right now, but for now, the centrosomes, they are producing some spindle fibers. So the way you draw it is you can make line going in between those, and they don't have to merge, but they have to start from the centrosomes. So the spindle fibers are growing. That's the second thing. First thing, the nuclear envelope disappears. Second thing, the spindle fibers are getting ready, produced, and get out, right? The chromosomes are completely condensed. So that's the third thing, because during G2 it's not necessary that all the chromosomes are completely condensed. But right now the chromosomes are completely condensed. Remember, I only use one chromosome for this example here. So that's your prophase. The nuclear envelope disappears. That's one. The chromosomes condense completely. That's two. And the spindle fibers form. That's three. Okay, let's switch to the next phase, which is going to be our metaphase. Metaphase is that step where all the chromosomes are attached by the spindle fibers and align in the middle of the cell. So we're going to make a cell here. So during our metaphase, now we don't have nuclear envelope anymore, right? The centrosomes are placed on either side of the cell. That's their job. They're going to be the guides. So when you draw the centrosome and the spindle fibers, make sure you draw them going to the middle and not further. And they don't have to reach each other's. They usually don't quite yet. And the chromosomes are aligned in the middle. So in this case here, I'm going to make my only one chromosome I have. And they align as such in the middle of the cell. That's metaphase. When you do those, remember that the spindle fibers attach to those. So you can extend the spindle fibers on either side to the centromere of the chromosome. The reason for that is after that it's going to pull on it and pull the chromosome sister chromatids from that chromosome apart, each one on each side of the cell, right? That's really important to realize that that plane here, it's in 3D, and here I have only one chromosome, but keep in mind we have 46 chromosomes total, right? So when you align them all in the middle and pull them apart, you're going to pull 46 chromatids on either side. They all replicated, right? So you have 46 chromosomes that makes for 92 molecules of DNA because it's already replicated the chromosome, right? So your metaphase is now done. The next phase, which I will start back down here, why not? The next phase here, it's anaphase, and that anaphase, it's really when the chromosomes are pulled apart. So what do we mean by that? If you make the drawing of your phase, make a cell right there. Still no nucleus, right? Your centrosomes are on either side still. You can barely see that one. Okay, here. They still reach out with spindle fibers. And I'm going to make one of them shorter, and you'll see why. I'm only drawing one chromosomes, but there are 46 in there. And now you have 92 chromatids that are pulled apart. And what we mean by pulled apart is, remember that centromere that was in the middle of the chromosome, that's where the spindle fibers attached, and they pulled apart. So that fiber pulled toward that centrosome, that fiber pulled toward this centrosome, and now you have the two chromatids going apart, and they're folding their arms like that. Because imagine you attach a bundle to my waist, and you pull really hard on it. My legs and arm will fall behind, while my waist will be pulled really fast toward you, pulling on the rope. So really, that's the key to remember, that the chromosome legs are lagging behind, dragging behind, while the centromere, is pulled toward the centrosome. So that's your anaphase. Anaphase is when you separate the sister chromatids. It's a really important part of mitosis. Now we go to the telophase, that is the last step of mitosis. Technically, cytokinesis is not in mitosis. So the telophase is the kind of invert of the prophase, if you want to think about it. So as we had three important things that were happening in a prophase, we have three important things that are happening in telophase. In telophase now we have two bundles of chromatids, right? So I only drew one chromosome from the start, so I'm just going to put on either side the sister chromatids from the same chromosome on either side of the cell. Now, importantly, you have a nuclear envelope reforming on either side of the cell, right? The centrosomes, are on either side of the cell, right? So during that phase, there is duplication of those centrosomes and nuclear envelope reforming, and the chromosomes that are uncondensing. So it's really kind of the opposite from prophase. The spindle fibers are resorbing as well, so now they're really short. It's kind of the cell going back to the previous state. The last and really important phase, the cytokinesis, it's not really part of the mitosis, but I'm going to draw it this way. And here in the middle, that's the cytokinesis process. It's when there is a pinch that occurs on the cell, that pinches on either side. That pinches on either side of the cell until it closes and makes two different cells. And of course, on either side, you have everything from centrosomes the nuclear envelopes are now completely reformed. So there's only the nuclear pores. And the chromosome is uncondensing, so it's not extremely clear anymore. It's kind of more going back to that yarn state. And that cytokinesis is the end of the phase that is not interphase. So cytokinesis marks the return to interphase for cells. They actually now have two daughter cells from the initial parent cell. And this is your mitosis. So once you've done it, you need to make sure you go back through it over and over again, redraw it. My advice is always the same thing. You draw as much as you can remember from memory, like I did here. But then when you cannot put anymore, you put a timer, let's say like 5 minutes, then you go back to your notes and add to it, correct the mistakes and all with a red marker or red pen. Then you put it away, and an hour later, you do it again from memory. So you don't look at your notes or what you've done before, you do it again. And once you've done that like 5-6 times, you will see that every time, every iteration, you will fill back in, you will correct less and less mistakes. And at some point, you'll be able to draw it from memory, the whole thing. And once you can do that, you know mitosis. And it's really important that you get mitosis down this week, as next week we do meiosis. And although similar, it's different. Okay, this was mitosis.